Welcome to iLinks Genomics Data Analysis Portal. iLinks, or Integrative Links Data Analysis Portal, is a user-friendly web tool for leveraging links perturbation signatures in the analysis of genomics data, as well as for mining links perturbation signatures themselves. In this introductory video tutorial, I will describe the high-level structure of the portal and the analysis that it facilitates. Let me first start with the conceptual structure of the portal by going through a few PowerPoint slides. Then I will give a quick tour of the portals. Other video tutorials and PDF documents describe each analytical pipeline in the portal. So the portal facilitates analysis of molecular signature of cellular activity. So let me first define what these are. So signature in our case is a list of scores or activity levels for a list of genes or for all genes in the genome at a time, in which case we refer to them as genome-wide signature. Currently supported signature types are differential gene expression signatures consisting of log ratios of average expression levels between different sample types, and transcription factor binding signatures consisting of transcription factor binding scores and probabilities derived from ChIP-seq data. The portal facilitates searching of pre-computed genome-wide signatures residing in the portal backend databases, as well as constructing new signatures. New signatures can be constructed using genomics data within the portal, or they can be uploaded to the portal. There are currently 5 libraries of pre-computed signatures in the portal. The first library are links perturbagen signatures, which are transcriptional signatures of chemical perturbagen activity constructed based on Broad L1000 assay data. The second library of the signatures are links knockdown signatures, which are transcriptional signatures of shRNA gene knockdowns. Then we have disease-related signatures, which are transcriptional signatures constructed by analysis of GeoGDS collection. And then ENCODE transcription factor binding signatures, which are transcription factor binding signature constructed using ENCODE ChIP-seq data. And finally, the legacy connectivity map signatures contain transcriptional signatures constructed from CMAP2 dataset based on Affymetrix platform. Raw genomics data that can be used to construct new signatures within the portal itself consists of more than 3,000 public domain genomics datasets. This collection includes GeoGDS datasets for human, mouse, and rat. It includes TCGA RNA-seq data, and then a list of selected custom curated datasets of interest. In general, analysis flow starts with finding, constructing, or uploading a signature of interest, then identifying pre-computed signatures that are concordant with the signature of interest, and finally establishing the patterns of gene activity scores that are responsible for this concordance. Here is the quick example of what this means. The search of ENCODE transcription factor signatures with the keyword ER-alpha returns a list of 5 signatures. By following the link with the signature's ID, all concordance signatures for these specific signatures in different libraries are displayed. I can now perform a group analysis of concordant signatures. Here I selected a few connectivity map signatures corresponding to ER-alpha agonists and antagonists. Finally, the analysis of differential expression scores of these signatures identifies groups of genes, and there are patterns of expression that drive these concordances. I will now switch back to the portal itself and give you a quick tour. To start your analysis, you can select one of 4 different pipelines for finding or creating a signature on the left-hand side. The first default pipeline is to search within the libraries of pre-computing signatures. Hovering over the names of individual libraries will display quick descriptions of the signatures contained by the library. If you want to just browse all the signatures, you can hit the search button without putting a search term. If you do that, so first 5 signatures from each library are listed, and also the number of signatures contained in each library altogether are displayed. So there are close to 85,000 LINX gene knockdown signatures. There are around 29,000 LINX perturbagen compound signatures, 4,300 disease-related signatures, 494 ENCODE transcription factor binding signatures, and finally 519 connectivity map signatures. If I now go back to the beginning, I can recreate this ER alpha search that I showed you in the slides by typing ER alpha and selecting only ENCODE transcription factor binding signatures library. If I hit the search, it will display the first 5 of total of 6 signatures that were identified. If I select all records, then all 6 signatures will be displayed. The second pipeline for creating a signature of interest is the upload pipeline. So if you select or follow the link of the left-hand side, it says upload, you will be given an opportunity to upload your own signatures. And the example of signature is here. So signature consists of a list of gene IDs and scores, in this case log ratios, and associated p-values. Going forward, the third pipeline is the pipeline that allows you to create a new signatures using one of the data sets that are stored in the backend databases of the portal. Again, there will be a separate video that describes how exactly you use this backend genomics data to create a signature of interest. And finally, the last pipeline is designed to identify signatures of interest by performing enrichment analysis. In this case, user supplies a list of genes, and the enrichment analysis of this list across all signatures, pre-computed signatures, is performed to identify signatures in which genes in this list are unusually highly differentially expressed or unusually highly likely to be bound by a specific transcription factor. Finally, there is one more way to start using iLinx portal by searching for groups of concordant signatures in this meta-signature analysis. Again, a tutorial video will be posted here that will give a specific example of how this particular analysis pipeline can be used to identify groups of concordant signatures and perform the follow-up analysis. This concludes the introduction to the iLinx Genomics Data Portal. To acquaint yourself with the specific pipelines and see examples of how a portal can be used to identify interesting associations, please refer to other tutorial videos that are specific to each analytical pipeline.